Hi everyone, and welcome to the 12th TIA Centre seminar of the academic year. So my name is Adam Shepherd, and I'm a postdoc here at the TIA Centre. For those that are new to our seminars, we invite researchers and leaders in the area of computational pathology and associated areas to discuss their work. And today I am delighted to introduce Dr. Jinxi Xiang. Jinxi is a multidisciplinary researcher specializing in signal processing and machine learning for healthcare applications. He integrated machine learning with medical imaging during his doctoral study at Tsinghua University. At Tencent AI Lab, he developed AI tools for clinical pathology using the techniques of image video coding and multimodal learning. The title of Jinxi's talk is A Vision Language Foundation Model for Precision Oncology. Jinxi, thank you very much for joining us. And whenever you're ready to start, please feel free.
Great. Thank you. Thank you for inviting me. So I would like to share a new study that was published in January by our group. Is a vision language foundation model for precision oncology, and the project lead of this study is Professor Rizhang Li. So as we all know, the clinical decision-making process is actually a multimodal information integrated. So for texts, that could be included publication, clinical notes, and all other text information in clinical scenarios. For images that could include pathologist images, CT images, and MRI images. So especially for oncology treatment, we are interested in incorporating clinical note and pathologic characteristics, which is derived from the pathology images. From our observation, we can see there are many complementary characteristics of pathology images and clinical notes. First of all, pathology is rich, raw visual features that was obtained from imaging devices. So this pixel information is derived from the imaging device is not processed by human being. And on the other hand, the clinical note is processed information actually from expert analysis. So I will give you some example here. First one, in clinical note, they might contain some diagnostic information that were not shown by the pathology images directly. For example, it might write some information about the differentiation, or if you have some testing results, such that the PD-L1, the TMB testing result might be involved in the clinical note as well. And secondly, there's some prognosis information. So these are some some initial conclusion from the clinician, perhaps the stage of cancer, or whether this patient has central nervous system metastasis, such kind of things. These are closely related to the prognosis outcome. And more importantly, for treatment prediction, there might some important meta information, such as because for patient, especially in advanced stage, the patient will receive multiple treatment plans. So, and we can learn from the notes that whether this patient also receive concurrently chemotherapy or immunotherapy. Such kind of information are not shown directly by pathology image as well. So these two information are in natural, very complementary with each other. So our motivation is try to develop some kind of multimodal foundation model that can make the most of these two modalities. So why do we need to develop a multimodal foundation model instead of making use of the off-the-shelf to single modality models? There are some consideration here. So first of all, and there's some drawbacks of single modality model. What we call is domain gaps. This domain gap is caused by the feature space in the text input and image input. For example, we can encode a text input with an encoder, and the feature embedding will be in a text feature space. And for the images, we also have some image in image space. So this feature space are not aligned with each other in natural. So this misalignment can cause the model will disable us to retrieve images from text or retrieve text from images. And if you also have no support for zero-shot image classification, which is a very beautiful feature property for multimodal embeddings, because we would like to, when in the clinical scenario, only a handful or limited labels are available, we cannot train a supervised classifier in these cases. So for zero-shot image classification is also a very useful property for models. So the other limitation is that we will miss something what we call multimodal insight, because we cannot leverage the complementary information from both modalities because they're not aligned with each other. This could be a little bit sophisticated to understand. We can put it in a 2D feature space. For example, if the texts and the images are actually orthogonal with each other, and if we try to fuse them and add these two vectors, we only got a very marginal gains. But on the contrary, if we can somehow develop a model that can align both feature space together, then which means that the feature embedding of text and the feature embedding of images can understand now with each other. That will naturally bring two benefits. So this alignment of image and text will enable us to do the retrieval task and the zero-shot image classification task. Also, we can maximize the multimodal insight. And this in equivalent is to into this space would be you align the two vectors now on the same direction. So if you added these two vectors together now, you can now maximize the multimodal gains. But please note that the feature space in encoder is in a much higher dimension. So it is not a 2D dimension. So this example is only make it intuitive to understand that why we try to develop such kind of multimodal feature space to align these two modalities. So another motivation we try to develop a multimodal foundation model is that we try to do something called text supervised signals into the image representation. As we all know, and many pathology images foundation model nowadays largely rely on the self-supervised learning strategies such as DynoV2 is the most successful in this area. And that for DynoV2, we actually constitute some pseudo labels from image-only inputs. So such that we mask some patches of the input images or crop a local patches from a global one to ask the model to predict which patches are from the same input. So in this way, the feature encoder try to learn such kind of image representation in a reconstruction or discriminative ways, or from image alone. But for multimodal pre-training, the story is different. Actually, we try to align the image and text together. So in this way, text is used as a supervised signal. Let's take an example, what a text would be for an input and image text pair. So for this example, the text would describe the shape of the nuclear and the layer of the epithelial cell, and also it will describe the appearance of the tissues, and will give a short conclusion on the morphology of the whole patches. So this input supervised signal is very rich and very good for the image representation to learn from, because they are totally different from the pseudo labels from the input images. So in this way, we try to instill the text supervised signal into the image representation. This is our second motivation to use multimodal foundation model. So if we looking retrospectively, we should know how far are we from a multimodal foundation model, especially in the past year and the past few years, and especially 2022, there are many vision models that are trained with billions of image patches from millions of host-like images with around maybe approximately billion of model parameters. So they make us very remarkable progress in 2022. There are many honorable mentions, such as Virtual 2, Unique Gigapath, and HOPTS. These are all very robust and generalized image representation models. So, but on the contrary, we can see that the progress is relatively very slow. And to my knowledge, the first one might be the PLIP published on Nature Medicine in 2023, and later there are some following studies such as CUT 1 million and the CONCH in 2024. But compared with the vision length foundation model, there are many challenges, especially in the perspective of data collections. And we can only make use of around 1 million image text pair. This is a very small scale compared with the natural image text pair. For example, for lion, it is a very large natural image text pair, which has 5 billion image text pair. And even for CLIP, which is published like 4 years ago, it has around 400 million image text pair. But we can only make use of around 1 million image text pair. So this is one of the main barrier in this field, the data collection. So we will recap some study in this field, and we will understand why we tried to develop such kind of model and what contribution have been made in the past studies. And for example, for PLIP that was published on 2023, and as far as I know, this is the first vision length foundation model for pathology, and it is further fine-tuned based on the pre-trained CLIP, and it curated around 200,000 image text pair from social media, Twitter. At that time, they used the image post by pathologists that has a paired text description, so that they collect like 200,000 in training data. And after pre-training, it has a very good property, especially for zero-shot classification. So for zero-shot classification, for somebody who might not familiar in this field, is that we try to extract the input images, and the task is try to know what potential classes is this image belongs to. So, and the user can write the description of some potential classes in natural languages and encode the text so that we can get the image and text cosine similarity. And the final output is the text description, where the highest text and image cosine similarity. So in this way, it's very scalable and very flexible, and you do not need to train a model. That's why we always try to investigate this property of multimodal foundation model. And also there are many other tasks here. And some following study, especially Conch, made this represent a substantial leap in this field because they collect a much larger data scale from a private center, and with around 1.1 million image text pair. So if you further enable slide level classification and segmentation, and it make a very remarkable progress in all patch level benchmark as well. So in conclusion, we can see that there are many bottlenecks in vision length foundation model compared with the vision only model. The first one and most notable one is the scarcity of well-annotated image text pair. And this cannot be achieved by collecting some social media image text pair, because the scale is only limited to millions of pairs. And what we try to do is that we try to leverage the unpaired data, because we all know the unpaired data are readily available in clinical texts and public data source. So if we can first make the best use of unpaired data, and that could be alleviate this problem largely. So the second bottleneck in this field is that actually there are many benchmark for multimodal foundation model, especially there's some image text retrieval or the resolution classification. Although this is a very important task, but there are many tasks that have not been investigated, especially we do not know how we can make the best use of a complementary probability of an image text pair for more complicated tasks. And this leads us to the third consideration of this model, is that the current applications are validated on cancer detection, diagnosis. Although these are very important tasks, but the pathology has already made a highly accurate diagnosis and detection using the image alone. So, but for their more challenging tasks, are following the detection and diagnosis, which would be the treatment, the prognosis. This task would require the model or require the pathologists to integrate as many input information as possible so that they can explore more translation value of the meta model, and instead of testing the model on benchmark datasets. So in conclusion, our study will bring some advances in this field. First one will be the conceptual advances is that we try to integrate the complementary information from image and clinical note reports, and it shows a superior outcome prediction over either modality alone, which means we're using pathology image alone or using clinical note alone. The second one is the translational advances. It is that we evaluate the performance on real-world datasets, such as the melanoma relapse prediction, cancer prognosis, and some very challenging immunotherapy response predictions. And last one is that the technical advance, as we mentioned before, we use unpaired data, and we try that was integrated into the same encoder framework and include some pathology-specific adaptation in this field. So we walk through all this detail in the following slide. As for the first step, which is the pre-training, and we try to use around 1 billion text token extracted from 1 million pathology-related articles and around 550 million images images from around 13,000 slides. So these data are distributed across all common cancer types, and what is interesting that compared with the publicly available pair data, the first step we use the unpaired data and what a much larger scale than the pair data. So the first step we train for like around a week on multiple nodes, and we can break into the details of these first steps. Actually, we used two training laws. Both are mask laws. We masked around 15% of the input text token and 14% of the image text token, and these losses try to optimize the model's prediction over the missing tokens so that the model can understand the context of the images and the text simultaneously. So the network architecture is a little bit different from the standard vision transformer. And what's different, what makes it unique is that we try to use a shared self-attention layer and use a separate FFN layer. This architecture has been proven to be very successful in other fields, such as Bayes-3 stable diffusion or the MOE in the large language model, which also use separate expert for different using scenario purpose. So for pathology adaptation, and we try to leverage the property of whole slide images, which is the multi-scale image with when we try to zoom in with a large magnification, we can see the morphology of the cell and the nuclear, and we zoom out, we can have more context. So we try to mix the magnification during the training, and we also use some pathology-specific staining augmentation, try to stimulate staining variation. In the second pre-training step, after we use all the unpaired data to pre-train, is that we try to use a handful, around 1 million image text pair, try to align this further. So this step would be very fast compared with the first step, because the data scale is small and we only need to train around a couple of hours to align these two features. Because although after the pre-training of the first step, although we have very strong representation of both modalities and they are unified in the same model architecture, they're still not aligned with each other, but they're ready to do so. So we only use 1 million text pair to align for a couple of hours trainings. So during this second step training, we use the standard contrast loss with an auxiliary loss with fine-grained alignment, because we all know the contrastive learning is try to align the class token of two modality, so it is a global alignment, but it cannot guarantee fine-grained local alignment because there are many features in the visual space and also in the text token space. So we try to use another fine-grained alignment text along with the contrastive loss. During the second step training, we do find some challenges here. The first is that since most of the public available image text pair are collected from the internet, so such as the YouTube, textbook, or social media, so it contains lots of noises. So the first step, we train our model on all the raw data, including noisy data, and then we will score the training data text pair after the training, and we will see the distribution of the image text similarity here. And on the x-axis, we can see is the image text cosine similarity score, and y-axis is the count. And we can see that there are many noisy sample probably below 0.1 threshold, and we can visualize this noisy sample and see what happens in this sample distribution. We might see that there are some unmatched image text pair, which is very common for internet crawled dataset. And we can also observe many low quality image data, such as blur or the misfocus. So we will eliminate such kind of noisy samples because we do not we think this will harmful to the model training, and we will eliminate such kind of noisy data and then retrain the model with the clean data so that we can improve the model performance a little bit further. Compare with the since I have proposed many adaptation and many pathology consideration in the model, we also do some ablation study compare with the off-the-shelf to use pathology images without all this adaptation here. So we can see these adaptation are very important to make essential improvement in the final downstream task. Okay.
Sorry, can I just ask a quick question before you go
Okay.
forward? Yeah, when you retrained the model on the clean data, did you train from scratch or did you
We train from
update the one that? Oh, okay, cool. Thank you.
yeah, because we think this noisy data does not make any contribution to the model. Actually, it will degrade the performance. So after the filtering, we will retrain the model from scratch. Actually, from the stage 1, which is from scratch, that's on the sixth step 2. Okay. And after pre-training, the model is flexible to use in many downstream tasks. For pathology, we can roughly divide it into two kind of downstream tasks. First, perhaps is the patch level dataset. There are many public benchmarks, so we will do the benchmarking for the first category of downstream task. And the second category is the slide level outcome prediction task. We think this one is much more exciting, and because we collect data set from the real world scenarios and evaluate the model on slide level. So, and it might have some potential reference for the clinician to use in the future. So the first one in patch level benchmarking is the text image retrieval task. This task is simple to use because we extract the embedding of the images and embedding of the patch of texts. So, and there are a pool of patch candidate and image candidates. So given a query of images, we can use the embedding of the images to calculate the image text similarity and try to know which text match the image query image perfectly. So we can show that the improvement of the image text retrieval made here by our proposed model, and this image text similarity score here, and we highlight some text description that actually shows the visual features of the query images. The second one is the visual question answering task. Actually, this is a very interesting task because and we will show the model and images and ask them some question based on the images. So the input would be a multimodal input, and the model try to make a decision based on the input of the image and text. And to simplify this task, because we try to evaluate the multimodal ability of our models, we do not make actually the generative, but we actually make it a discriminate task as the output. The dataset we use a pathway-QA dataset, and here we show the performance of our model. And the third one is the image classification. This is a very classic task because we use it every day, and especially there are two label efficient tasks, which is zero shot classification and few shot classification. Zero shot classification, as I mentioned before, is you do not need to further train the model to use it. You just describe your potential classes into natural languages and calculate the cosine similarity and make the final decision based on the similarity score. For few shot is that we only use a handful, maybe 1, 2, or 10 label of each class, and then train the model in a light way so that it will have better performance. This is very important for maybe rare disease diagnosis in the clinical scenario. So this benchmark has very great clinical values here. And moving forwards on the patch level benchmarkings, we try to make a big step, try to make a further step on the slide level outcome prediction task. And because diagnosis is the primary step, perhaps the first step throughout the whole oncology treatment pipeline, following diagnosis, we will have some treatment and some prognosis evaluation challenges. This all required us to make the full use of the multimodal input and devise a model that is robust and generalized to different application, that was not limited to only one or two cancer. So the first one, first of all, we use a unified framework for all the real-world outcome prediction task, and which is we will pre-process the whole slide images and try to segment and crop them into a small input patches and extract feature and aggregate this feature to get a global embedding. For the text input, the pathology report usually will have like thousands of words, so that would exceed the context length of the MUSK language model. So we will pre-process the input pathology report with a large language model and make it a structured report. And the prompt will be designed across by the specific downstream task we use, so that for the structural report, we can use the MUSK model to encode and get the text embedding as well. We can fuse these two embedding and make final prediction by changing the final prediction layer for classification or prognosis. So this is a unified and flexible framework for all types of real-world data set. The first one is the melanoma relapse prediction. Melanoma is a cancer of skin that is derived from cell responsible for skin pigmentation. And this data set contains around 1,342 patients. And the text input would include the patient's ages, sex, prime site, and medical history. These are very closely related to the final relapse prediction, and input images is a routine H&E host line images. So after training, the model will have very good understanding of the both input and of text and images. After only trying to train the lightweight classifier, we can get the performance of the melanoma prediction task. And as the show, the AUC curve and the sensitivity we evaluate here is very promising. And on the bottom line on the right side of the slide, we show the model can use report-only input or image-only input. But compared with the report plus image input, which is multimodal, the performance will degrade to different degree. We also try to interpret what we can get from the attention score of the MUSK model and try to understand why the model made such kind of prediction. And on the left column is the input host line images, and the second column is the heat map obtained by the attention score in the MIL classifier. And as we zoom in and see there's some clinical pathology features that is closely related to non-relapse and relapse cases. For relapse, the ulceration of the skin is very typical characteristic, and there are some lymphocyte activities as well. So we invite some pathology to make some description of the ROI of the non-relapse and relapse cases, and they found some evidence that is consistent with their experience. And for the second task, we try to do a pan-cancer prognosis prediction. This cancer type cover the most common types across the whole body. And the input is the clinical report again, and the input images is the H&E host line images. And for the input pathology report, as we mentioned, is a raw unstructured report with thousands of words. So we cannot just feed in the raw report into the MUSK model, and because it will exceed the context length of the model. And they also have many redundant information that has no relationship with the final prognosis prediction. So we try to design some design prompt to extract the information that's closely related to the prognosis, and also is to make the prompt universal for all cancer types. So what we decide here is that some tumor site description, differentiation, the evidence of lesion invading or other organ or tissues, and the lymphocyte metastasis that might be also described in the report. So after the pre-processing, we get a structured report here. If we describe in around one or two hundred token, one token, around one token, we can get all this answer of the prompt that was closely related to prognosis instead of using the raw unstructured report. So after evaluating across all 16 cancer types, we can see a conclusion that the multimodal output, which use pathology images and report, consistently outperform the image only or the report only when we try to use the C index as the final metrics. Here we show the KM curve across 16 cancer types. So for some cancer type, the model performed especially well. And as we see, for example, for RCC, but for some cancer type, the model struggled to do that because all concurrent models struggle to do such as the GBM cancer type. So, but okay, I see a question here.
Yes, just a quick question. I wonder, what would you expect to happen if you did not convert pathology reports into the structured text that you did? What would happen, do you think, that if you use the text directly extracted from pathology report?
Okay, so as far as I understanding, your question is that try to put the raw text report into the model instead of doing the pre-processing, right?
Yes, exactly.
Actually, since the context length of the model cannot accept thousands of words, so the first thing is that it will it will clamp. It will have only, like, if it only had 200 words, so the remaining words cannot be encoded by the model. Or you can crop the text input into many segments, but it will lose context to understand that. This is one drawback of putting the raw input to the model directly without pre-process. The second one is that there are many redundant information. There are many information that's not closely related to the final prognosis output. So the model will struggle to understand which tokens are important for the final prediction. So since for the prognosis task, the training label is only very limited, like perhaps 100, or perhaps for some tasks there might be thousands, but the label is very small. So for such kind of high dimensional input, the unstructured raw report is high dimensional input. The model will struggle to understand which input closely related to the output. So these are two drawbacks of the not pre-processing the input report, text report. So in most of the case, it might fail because the limited label cannot drive the model to extract very useful text input from thousands of words. But in comparison, if we only have 100 or 200 input text token, and all these text tokens are closely related to the output, the model can converge very quickly and will have a very good prediction abilities.
And may I ask a follow-up question to that? Oh, sorry, Mr. Wei, you continue there.
No, sorry, just wanted to ask if you had any challenges with some missing information in the reports because you want to form a structured report out of the raw report. Say if some report has ICE information, but the others don't have it, how did you handle that?
Yeah, that's a good question. Actually, we for some prompts, not all report can have six all these 1, 2, 3, 4, 5. Not all report have these 5 answers. Perhaps for some missing question, they will just put an unknown here. So the model can actually they know we can derive from all these 5 prompts, but some prompt might have missing output. So for example, if they have no lymph node metastatic information, it will just put a description that there's no lymphocyte metastatic information shown in the report. So the model can understand this language description.
Has that answered your question, Mr. Wei?
Yeah, it did. Thank you very much.
Fantastic.
Thank you.
My follow-up question was going to be very similar, to be honest. It was about how we ensure the accuracy of these structured reports. Especially, how do you, for instance, stop ChatGPT from hallucinating in the actual responses, in the generated structured reports, sorry.
Yeah. Actually, yeah. We use GPT to generate the report by using this prompt. Yeah.
Yeah. So, I mean, my question was, I think maybe in the interest of time, maybe we'll go to it at the end.
I guess in the case of summarizing stuff, ChatGPT doesn't
not
really
pretty good.
listen much anyway, does it?
Yeah.
Okay, we
Thanks,
are
Jinxi.
reaching the end of the slide, so we can discuss that question further. No problem. And the final task I'm going to present here is the lung cancer immunotherapy prediction. And for this immunotherapy prediction is very challenging in clinical because for some FDA-approved biomarkers such as PD-L1 expression or TMB, it only have very limited predictive power, like around 0.6 AUC in practical real data. So this is a unresolved question for a long time. And our ambition is try to use MUSK model to find out the responder, and typically for lung cancer, it would be 20% of lung cancers, so that we can dismiss the patient from the clinical, the financial burden and the toxic side effect, and try to precisely find out the 20% responder. And again, so we will also pre-process the input. And for this task, we collect some lung cancer data from the Stanford Hospital. So in our center, they contain lots of information, like always exceed thousands of words. So, and these prompts are designed by the clinician, and they think that's might closely related to the immunotherapy prediction, such as the differentiation, the lymphocytes, the PD-L1 diagnosis, such kind of things. I mean, also has an example output like this. So this is the text our model is going to encode and get the text embedding. And the final object response, which is actually a binary classification, and it varies in terms of AUC, is around 0.76 or 0.73 around this interval. And also another task is what we call progression-free survival. So the evaluation metric is a sig-index here. And we also do some subgroup classification. And we can see for all the patients here, the KM curve can stratify the high risk and low risk patient. And for the PD-L1 treatment, they are PD-L1 equal to 0 and larger than 1. These are two subgroups that we also try to understand whether the model can have better understanding of their different patient characteristics. And EGFR is also very important gene mutant in lung cancer. So we also further carry out some multivariate cohort regression analysis on all patients and trying to find out whether it is a significant predictor over other commonly used biomarkers such as the CNS metastasis, EGFR mutant, will also great contributor in the prediction and compare with other histology and PD-L1 predictions. Okay, the final slide as the same is the visualization that we overlay the heat map on the whole slide images. So actually there's no very clear clinical guideline compared with the melanoma prediction, because there's some guideline for melanoma prediction, relapse or non-relapse. But for immunotherapy, responder and non-responder, there's no such kind of guideline. So, and we try to explain the discovery of the model and invite some pathology to write some description whether this model actually is learning something instead of making random guess. So, and we write some detail description over our region of interest, and we should also show very detailed in the paper. So, there's some might be in the responsive cases. Responsive case shows some high lymphocyte inflation and minimal stroma, while the non-responsive case might show the opposite. So these are my presentation here. So our code model and our more open source available. Please give us if you like. Thank you so much.
Fantastic. Well, thank you very much, Jinxi, for a really great talk. And with that, I suppose we'll open the floor to questions. So does anyone either online or in person have any questions they'd like to start with?
Yes.
I
think that was fine. Don't worry, please go for it, Mark. So we've got a question from Mark.
I was just wondering, I think that in the vision part of your training you were taking images at multiple resolutions. I was just wondering, sort of during inference on the various tasks you were trying it on, what resolution did you find worked best for you for your model?
Okay, yeah. There's a training detail. Actually, for different tasks, and we use actually at 10 10x magnification for most of the cases. So we do not tune these parameters, but we think this at this magnification, they might have better understanding of both the detail and the context. And yeah, so that's the default setting. And without further explanation, is the default setting for most cases.
Okay, thanks.
Thank you very much. And a question I had on your slides actually. So you obviously, of course, you found that by including data that was a bit too noisy, that it resulted in worse performance generally. Did you then see how it worked afterwards if it was inferred on noisy data? Was it very, very susceptible, I suppose, to noisy data in general?
Okay, so for the, you mean the noisy data?
Yeah, exactly.
Okay, yeah, actually we do some, why do we make such a filtering process is that we do some ablation study and show with the noisy data and without the noisy data, the final version of the model will have different performance on the downstream task, especially for the retrieval. Yeah, and it actually shows improvement after eliminating such kind of noisy data. That's why we decide to use such kind of filtering process.
Okay, no, brilliant. Thank you.
Yeah,
that makes sense.
Yeah.
And, so
I guess a follow-up would be, how did you choose the 0.1 threshold that, would increase in that, clean your data even more and perform even better?
did you get that question? Sorry, Jinxi.
Yeah, maybe there's some information lost through online conferencing. Could you please rephrase it a little bit?
Yeah, so George asked, how did you come up with a threshold to remove the noisy samples?
The threshing hour.
The threshold to actually remove the samples.
Oh,
So
okay.
it's 12%.
Actually, this is an empirical number, so there's
Okay.
no specific reason. We might tune this number a little bit further by visualizing the sample. So if we put this threshold a little bit higher, we lost some training sample. We put it a little bit lower, we cannot eliminate some noisy sample though. But this is an empirical number. We can visualize through this image text pair and make the final decision about 10% perhaps is a good threshold. Yeah. Thank you.
Fantastic.
Yeah.
Thank you. And I suppose my next question is, so this is fantastic. We're seeing some really great foundation models now in general in computational pathology. So what's the next step, I suppose, towards clinical deployment? How do we use these foundation models now to actually help patients, or how far off this are you, or is your work?
Yeah, actually, there might be two directions. So since for the model pre-training part, although we use some unpaired image and text data that alleviate the scarcity of well-annotated pair data, but there's still a long way to go compared with the natural image text pair. And I think the model performance can be improved further if we have made great effort into the data collection. So this is one direction to improve the performance of the pre-training model. On the translational side, we also try to collect more multi-center data in clinical practice so that we can validate the performance across different population, across different regions, so that it will have true translational value for the patient. Yeah.
Thank you.
Thank you.
And do we have any final questions from anyone online or in person?
I have
No
a few
more questions?
questions if no one has any question.
Please go for it, Mr.
In the second last slide, I've seen like you have shown like slide wise interpretation based on like what text has been generated. So since it was slide level, do you see that consistent across different outcome predictions? Like I think it was recurrence versus I think it was in the second last slide when you were showing slide level results. I think this one. So in the top one, you're showing something that came up with responder and non-responder in the bottom one. Do you see that consistent across different patients, or like do you see different text that is being generated for different images from the responders?
Okay, so for the text interpretation, and that's a little bit different. So compared with the visual images, and we do not correlate the text description for visualization. And at that time, we do such experiment. But yeah, your suggestion shows a good way to understand the model about the text.
Yeah, thank you. And I also wanted, I think you might have covered that, but I might have missed about the pre-processing of the text while training the model. So do you need to do that manually? And like if a different person is gonna look at the clinical reports to for pre-processing, will it be different by different physicians or different experts?
Actually, the prompt is designed by some lung cancer immunotherapy clinician. So after the prompt design, we use this prompt to tell the large language model, which is GPT-4, to automatically curate the clinical report, not manually. So I would say only the first step, which is the design of the prompt, will require manual labor, and the following steps are all automated.
So you have a set of questions that you would like to look up for that information, but what if you have missing information for some of the cases? How do you deal with that?
Yeah, actually, and there might be this is a common question. And we will only, for example, if we do not have the information of whether the patient received other treatment concurrently, and then the answer would be there's no such kind of information shown in the report. And we try to make the model robust also to the unknown information, so that as we know, yeah, during the fine-tuning, they might extract information that make the best contribution, make the most contribution for the final prediction. So it can accept missing information by using a language description here that it is that there's no such kind of information. Yeah.
That makes sense. Thank you.
Yeah, you're welcome.
Thank you, Rachika. I think we've also got time for one last question, Mustafa, if you do want to slash your question.
Yes, just a couple of really quick questions. First of all, thank you very much for the great presentation. It's an amazing piece of work, lots of experiments, lots of technical details, perhaps lots of questions about them too. So I'm not going to go there. I'm going to ask a couple of questions that are not explained in the paper themselves. This is more of a questions that you might not find in other seminars, right? So for example, I'm curious to know what was the most challenging part of the project for you?
Okay. Yeah, actually, it's the first part of the pre-training, because when we try to launch this project, we follow the study. There's so many good stories, especially Philip and Conch. They curated image text pair to improve their performance, and this is their direction. But at the time we try to carry out this study, we do not have sufficient labor to curate image text pair, and our performance was stuck to a level that cannot progress anymore. And after that we try to use a unified architecture with unpaired data, and then further improve the performance of the pre-training model. So that would be one of the first challenges of the project. And the second one is that we try to investigate what's the true value, especially translation value, except for the public benchmark on patch level, such as the image text retrieval, serial classification. So the second step, we try to investigate the translation value, and that's why we design and collect the immunotherapy prediction task, the pan-cancer prediction task. And it takes us a long time to collect these liver data sets, especially the immunotherapy, because they are very scarce to collect, very hard to collect. And also we need to curate it, all the clinical reports. So that also made a hard time at that during the development. So it has two challenges on the
Makes
model
sense.
development
So
and the
data
technical
was the challenge. I suppose it is,
Yeah.
yes. And we are very short on time, or ran out of time, but just one more quick question. If you
Okay.
go back in time, what would you do differently? Or in other words, if you want to improve the current work, well, technically speaking, how would you do it?
I would say I would collect perhaps more images data and more text data. So especially the pathology image data. So we rely on the open source data to train the whole model. And actually, as we can see in vision only foundation model, to collect many private pathology image as well. So if we got another chance, we might think this is a good direction to improve the model further if we have more private image data set to train the first step of the model. Yeah.
Well, amazing. Thank you very much. Hopefully one day you can modify your tool a little bit so it can be used to extract more data with it easily. But thank you very much. Thanks, Adam.
Thank you.
Well, thank you very much, Jinxi, for a really, really interesting talk. I think with that, we're going to have to draw the seminar to a close. Just to remind everyone, so the models are available on GitHub and on Hugging Face, right?
Okay, great.
Yeah, perfect. Right then, well, thank you very much, everyone, and hope everyone has a good rest of the day. And Jinxi, thank you again so much for a really fantastic talk.
Thanks for having me, Adams. Thank you.
Thank
Bye,
you.
everyone.